Hi everyone, good evening. Good evening everyone, welcome to the class. I'm really sorry for the delay, but all of you are here and I'm also here now. Good evening everyone, how are you all? Good evening and welcome to the 9th and 10th channel, right? All of you are the achievers and I'm your teacher Ankita. And in today's class, we will be discussing about the NCERT, right? The fundamental unit of life, of course line by line, we'll go through the textbook and of course we'll be reading the textbook. Interesting, right? If you can see me clearly and if you can hear me clearly, quickly give the thumbs up or you can write in the chat box. Good evening Riya, Purna, Rajput, Jaising, Chitra, Pranjal. Hi, yes. Anaga, Ashraf, Devdas. Good evening. So good to see that all of you are here. Right, we know that what we'll be doing in today's class. So I'll just give you some time. If you're not sitting with your textbook, go quickly now and get your textbook right of 9th class. And we'll start a session. Yes, good evening. Absolutely. Hi Tejaswini. Hi Epic. Hi Murali, yes. Riya. Good evening. Hi Ananya. Mona Lisa. Is it an echo? Is it? Do you have any echo? I think from our side, it's pretty much clear. It could be, or just check your phone and you can just refresh it. Yes, thank you for letting us know. Awesome everyone. So let's quickly start our session. It's part 3, right? Hopefully we'll finish this in today's class, right? Keeping our fingers crossed. Okay, let's start. Well, that's really very sad. Rajput, you're writing in the chat and you're telling that you're not able to see or write the chat. Interesting. Awesome. So everyone, before we get started, right, of course, you just had a session with Ankita Ma'am and of course it was all about T20, right? And of course, it has a very good connection with BYJU'S Scholarship Test, right? A few minutes back, we had a session with Ma'am, and of course, it was a quiz related to the cricket, right? And how these things are related. So, if you write BYJU'S Scholarship Test, you can actually win a chance to go to Australia to watch the World Cup live. Interesting, right? Super interesting. Apart from that, you can avail the scholarships also. So see over here, BYJU'S Scholarship Test. Go and register now. It's a very easy test. There are 2 dates remaining. The one which will be happening on 28th of August and then of course we have one on the 4th of September. Register yourself. The test is really very simple, super easy test, right? So round 1 is there and then of course once you qualify round 1, it's a very easy test. Of course it will be based upon the timings and the answers that you answer, and then of course we'll have the round 2 for it. Yes, so let's focus on round 1. It will be happening on 28th of August and on 4th of September. So register now and give the test. Apart from that, we have BYJU'S Mini Learning Program. Use the code YT first and you could be in the first 500 users to avail these amazing classes that we have. So it will give you an inside peek what happens in BYJU'S classes, right? So do try or check it out. Yes. Yes everyone. Epic is that yes, of course I'm in a big room. It's a very big studio that we have over here. Awesome. So everyone, I hope that all of you are clear with this, right? Now can I ask you a small favor, right? Now because of you all, we are a part, we are a family of 88k something, right? But of course we are planning that we'll achieve very soon 1000k, and it's not possible. But it's because of you, right? So help us reach this really very soon. So yeah, that's a very important thing we have. Yes. Now we'll be moving to our textbook. All of you now have your pencil, right? And have your you can have your textbooks also along with you, and here we are. Yes, we are starting it up. Yes. So I hope you can see the screen, right? Yes. Yes. Cool. Awesome. So of course, we will be using the pen and yeah. Yeah, it's working. Cool, right? Oh, awesome. So can anyone tell me where we left last time? Yes? Till now we have covered, right? Till now we started with the very first chapter. We talked about the fundamental unit of life. We talked about the cells. We talked about Robert Hooke. We talked about the experiments. We talked about the plasma membrane, cell wall, cytoplasm. We went into the details of cell membrane, right? Cell wall. And now we will be, yes, now we'll be discussing about the cell organelles. Now today is the last class for this particular chapter. We will be finishing this today for sure. So I want your cooperation, right? So here we are. We are done with nucleus also. We did in the last class. Yes. Cytoplasm is also done. And we will be starting with the ER, cell organelles. Okay? Everyone? Yes, very good Tejaswini. Yes, we'll be starting from the cell organelles. Now, let me see how many of you are here. And I want all of you to pay attention now. Yes? Okay, good evening to each one of you who have joined us, right, our class. I welcome you to the class. I'm your teacher Ankita. If you're just tuning in now, or if you're starting from here, very good. Very good. So, we will be discussing the cell organelles in today's class. We'll be reading from the NCERT textbook. Over here we have. Awesome. So, let's get started everyone. Now, it's a very, very easy topic. We have few cell organelles that we have in plants and the animal cells, and we'll be discussing about it. So quickly give me a thumbs up everyone, and we are good to go. Yes. Quickly everyone. Yes, Jay Singh Nahar. Yes, quickly everyone. Thumbs up or a smiley. Dipanshu, Kashish, Chitra, I can see. Nice, very good. Jai Singh, Shama, awesome. So we know that in every cell, right? Very good, Agna, Ashraf. In every cell we have cell organelles. Hobbies and craft. So over here, it talks about that every cell has a membrane, and basically it just keeps the inner cell content and outer the content that we have outside the cell in short, right? Like intact properly. So that's the first line that it talks about, right? Every cell membrane has around it and keeps its own content separate from the external environment. We have large and complex cells, including cells from multicellular organisms, needs to do a lot of chemical activities to support their complicated structure and the function. Yes, very good. It's okay, a big thank you for liking the video also. Right now, each of these organelles have different functions, and these functions are really, really very important. Right? They're really very important. So, these have different activities, right? And different kind of activities they perform. They make sure that the cell is performing properly. So, that's why they are really important. We know that we will see more of the organelles or majority of the organelles in the eukaryotic cells and not in the prokaryotic cells. I can see that many of you are asking that what are organelles? So the organelles are the small structures that we have, right? And we call them organelles, right? So for example, in our body we have different organs that perform a different function. Similarly, in cells we have these different organelles that actually perform the specific functions. Yes. Yes, actually you can write the answer, a question over here, but I would suggest that if you have one particular topic related to some question, you are asking what is biology? Oh my god, it's a very deep question. In a very simple term, is the study of the life that we have around us, right? Yes. Very good. So I hope that now we all are clear what are organelles, right? Now when we talk about the nucleus, in the last part of it, we know that it is a center of the cell, right? It controls various activities. Apart from that, we know that there are various organelles, and we are starting with the very first one, which is ER. Now everyone, we know that ER is really very, very important. ER is the endoplasmic reticulum. Now let's start with it. Yes. So the ER is a larger network of membrane-bound organelles and sheath. Now that's really important, everyone, right? Students, let's focus over here, right? Now ER is a large network of membrane-bound tubes and of sheaths. It looks like long tubules around, right? Oblique bags, like they're kind of a very, baggy structure, long, elongated baggy structure. Now ER, of course, there are two different types of it. We have the smooth ER and the rough ER. Rough ER looks rough under the microscope because it has the ribosomes attached on its surface. Very good, everyone. Yes, thank you for helping your friend out there. But I want all of you to focus over here. Come on, everyone. Yes, very good. SER and the RER. Now we are talking about the smooth endoplasmic reticulum. It has ribosomes and it actually helps in the synthesis of proteins. Right? It actually helps in the manufacture of the proteins and it sends to the various different parts. Right? It basically sends it to the different parts, of course, through the Golgi Apparatus. We'll be talking about that in a minute. Yes? So it manufactures protein and which are sent to the different parts. Then we have SER, which helps in the manufacture of fats and lipids. Very important, very, very important, right? And they're really very important for the functioning of the cell. Then these proteins and lipids helps in building the cell membrane. Again, a 1 marks question. I'm sure you would have seen this question in your examination about the membrane biogenesis, right? Yes. Very good. No issues. Welcome to the class. Yes, Tejaswini, biogenesis is nothing but a very simple process where the proteins and lipids will be helping in the manufacturing of the plasma membrane. Are we clear? Yes. So this process is called as the membrane biogenesis. Proteins and lipids help in building the cell membrane and we call that process as the membrane biogenesis. Some of these proteins and lipids function as the enzymes and hormones. And although we know the ER varies greatly, appearance in different cells, it always forms a network system. Are we clear? Everyone, are we clear with the ER? So in this one particular paragraph, we got all the information about the ER, be it the smooth and the rough endoplasmic reticulum. Lipids, Dave Das, of course, as they have mentioned over here, right, when we talk in terms of the molecules, different types of molecules are there, right? So we have carbohydrates, we have proteins, right? So lipids is also a type of, but of course it comes from the fats. Are we clear? Yes? Very good, Shama and Devdas have just explained. So I hope that you have the clarity. Yes? Very good. Purnima, I think I have missed your question. Can you repeat your question? And till that time over here we have the ER. Can you see over here? Smooth endoplasmic reticulum and the very rough endoplasmic reticulum. And over here we have the rough endoplasmic reticulum. Clear? Very good, very good. Now let's talk about, there are a few things again they have mentioned about it, which is that the one function of the ER is to serve as a channel of transport. Now this is important because many of us usually get confused. Okay, I wish I can draw it over here, right? So see over here, we have the ER, right? Okay, 1 minute. So we have the ER, right? And Harish sir, can we go back to the main, the slide bar? I'm just moving. 1 minute. I think I am over here. Yeah. Yes, everyone. Yeah. Thank you. So of course, see over here, we have this, right? I'm just making this ER not very proper, but I'm just giving you an outer structure. Wait, let's just make it a single line. So let's suppose, okay everyone, look over here. We know that we have the ER, right? And what it does, it will be making proteins and the lipids. Now these protein and lipids basically will be getting into the vesicles of small sac-like structures, and from there they will be moving to the Golgi Apparatus. Yes? So of course it plays a very important role in sending the material to the Golgi apparatus. No, Purnima, no. Lysosomes does not make proteins, right? It has the enzymes. It is not making something. Very good. Now we are moving back to the textbook. So here we go to the, okay, 1 minute. Here we have the textbook. Yes. So are we clear with this? Right? That what it does basically, there's one function of the ER is to serve the channel of the transport of materials, especially proteins, between the various regions of the cytoplasm and between the cytoplasm to the nucleus also. And the ER also functions as a cytoplasmic framework, providing the structure to give the various biochemical activities. Now this is a 1 marks question, which of course many of us usually miss in this particular chapter. In the liver cells, a group of animals called vertebrae like us, right? The smooth endoplasmic reticulum plays a very important role in detoxifying many chemicals, right? Poison and the drugs. Yes. Okay, I have taken your point about the tissues chapter. We'll have it. Thank you for letting us know. Yes, Dhruvi, yes, all of your points are taken. So everyone, can we quickly come back here and can we focus on the fundamental unit of life? Yes, we will have the one-shot videos on tissues for sure, right, the upcoming week. But are we clear with this ER, right? Very easy, and it's a very, very important part. I want you to all remember this. Right, in the liver we have more of smooth endoplasmic reticulum. And what is the reason behind it? Because they actually help in the detoxifying, basically breaking up and making the effects of poison and drugs very less in the body. Now comes the second most important organelle that we have is the Golgi Apparatus, right? Quickly, let's go through it. Detoxifying Devdas is basically toxic means poison, right? Very, very harmful for the body. So what these cells do, that break it down, detoxification, right? They actually break it down, they actually reduce the effect of it. Yes. Very good. Yes, I have taken your points on each and everything what you are saying. We will be coming up with these sessions. Okay, now focus over here everyone. The Golgi's body was first described by the Cambio Golgi. It's a very, very important scientist name. I'm sure most of you are kind of aware about it. Yes, Vidya Harshini, yes, they are same. Okay, Purna, bye-bye. Yes, very good, very good. So they consist of a membrane-bound vesicles, flattened structure, right? Again, a very important question which came in the Menti test and of course which we have asked, right? And they are parallelly stacked onto each other and of course they are called as the cisterns, right? This is important. Those parallelly stacked structures are called as the cisterns, and these membranes often have connection with the ER and they plays a very, very important role in the transportation, right? Yes. So the materials which are synthesized in the ER are dispatched in pack and of course it goes to the Golgi apparatus, and of course it plays a very important role in sending to the different parts of the cells altogether. The function is to include the storage, modification and packaging. 3 important functions: storage, modification and packaging. And in some case complex sugar. Now again a very important function of the Golgi apparatus. Complex sugar may be made from simple sugar in the Golgi apparatus. So the complex sugar which will be broken down, and from that, complex sugar will be formed, right, with the simple sugar when they get combined. And of course Golgi bodies plays a very important role in the formation of lysosomes. Now over here lysosomes, we'll talk about it, but over here we have this information on the Golgi apparatus. You can definitely read about it, right, about the scientists who discovered them or described them. Are we clear with Golgi bodies? Everyone, quick thumbs up. Yes, the chat got struck. I hope that all of you can see this. Very good. Awesome. Very good, very good, very good. So we are clear with this? I think that you were reading the textbook. Now we are moving to the lysosomes. You were asking about lysosomes. See how they are kind of interconnected. ER is making the proteins, right? ER is coming to the Golgi Apparatus, and from there we know that in the sac-like vesicles, in the sac-like structures only, we have the enzymes, right? And that's why we will be discussing about the lysosomes now. So lysosomes are membrane-bound organelles, right? Sacs, we can say, filled with the digestive enzymes, and these enzymes are made by rough endoplasmic reticulum. Enzymes are nothing but the chemicals, right? And they actually help in various processes. They basically make the process fast. Now, the enzymes, what types of enzymes we have? We have the digestive enzymes that will be helping in the breakdown of the materials. Yes? Very good. Now, lysosomes are kind of waste disposal system of the cells. Now, they are the one that will be helping in the removing the waste material. Now, these help us keeping the cell clean by digesting the foreign material as well as the worn out cells. Any of these cells, organelles that are not performing well, these lysosomes will make sure that they are not there, right? They will be destroying them. And if at all there is any entry of any foreign materials also, these lysosomes will make sure to break it down further or to digest it. Any of the food, foreign material like bacteria enters as food, they will be making it sure that they are not there, right? And lysosomes also contain powerful digestive enzymes, and they are of course capable of killing these cells also. Very good. Nice. Okay, I think I have missed some of the chat, but I think you are all are focusing. Let's focus over here. I can see there's a little bit fun there in the chat, but let's just focus over here, right? Protein synthesis and protein manufacturing. Shruti, is a very interesting question. So the protein synthesis occurs, right? Now we will be going back to the amino acids, and it has the relations from the inside of the nucleus and then of course the outside. Protein manufacturing is nothing but when the proteins are being made. So kind of a similar process, but a little bit difference is there here and there. Yes? But we usually call it as a protein synthesis, especially in terms of biology. We use the word protein synthesis. Okay. Now, are we clear with the lysosomes? Right? We call them as suicidal bags because if the cell is not performing well or if there's something wrong with the cell, what will happen? The enzymes will be released out of these lysosomes and the cell will die. And that is nothing but that's why we call the lysosomes as the suicidal bags of the cell. So when they are the protein synthesis, if we talk about a partial of it happens inside the nucleus, right? And when we talk about it, it goes further. Now we know that protein synthesis is definitely happening on the ER, right? We have these small ribosomes on it. So it has a connection with the nucleus, and these ribosomes are being made. The information that we have, it's basically coming from the nucleolus, right? We know that they are made there. So there's a little bit connection over there. Apoptosis, yes, killing of the cell. So we are clear with it, right? Right, everyone, are we clear with this? Very good. Yes, Vindhya, it's the rough endoplasmic reticulum. Yes, protein synthesis. But you'll be learning more further in your higher classes that where it all starts. Awesome, I can see lots of thumbs up. And yes, now moving to the mitochondria. We know it as the powerhouse of the cell. A very common name, right, is the powerhouse of the cell. And it has two membrane covering. Very important. The outer membrane is more porous. It was a question we asked in the Penti. We picked the question from the inside textbook, right, and over here we have the proof of it. So the outer membrane is porous while the inner membrane is not porous, it has deep folded. And these actually help us in increasing the surface area for the generation of ATP, right? And this energy generation of ATP is nothing but the adenosine triphosphate molecule that happens in the mitochondria, right? And of course, these ATP plays a very important role in helping in the performance of various activities, right? Lots and lots of activity that happens. We need energy, and this energy comes from this mitochondria. Very good. Adenosine triphosphate. How lysosomes came to know that a cell is not working well. Monaksha, what happens? There are a lot of chemical reactions happens in the cell, right? I'm just giving you one example of it. So in these chemical reactions, definitely something goes up and down. If there are so many bacteria inside the cells, or let's say there's some dysfunctioning that is coming from the nucleus, something is wrong with the cell. It could be lots and lots of reasons. Basically, if the chemical reactions are not happening well, the cell will come to know. They will try to repair it. But if that's not happening, they will not try to repair it, lysosomes. The cell or definitely the different other organelles will try to make sure that they are functioning properly. But if that doesn't happen, lysosomes will take the advantage of that situation and they'll make sure that they will release the digestive juices. Yes, very good. Yes, ATP indirectly we call it as the energy currency of the cell. Yes. Monish is asking why we call mitochondria as a powerhouse of the cell, because it's where we will see the production of the energy. Very good, right? So here we have the ATP, the energy currency of the cell, and of course the body uses the stored ATP, right, when it requires that energy. And mitochondria are strange organelles because they have their own DNA and ribosomes. That's why we call them as the semi-autonomous organelles. They can make their own protein because they have their own DNA and ribosomes. Are we clear, everyone? Yes, with mitochondria. Yes, very good. DNA is deoxyribonucleic acid, right? It's the genetic material that we all have. Very good, clear. Now moving to the plastids. A little bit similarity we have in mitochondria and plastids. Plastids also have first of all, they also have their own DNA, right? And they can make their own proteins. Yes. And of course, plastids are only found in the plant cells and not in the animal cells. We call plastids as a kitchen of the cell, right? As chloroplast especially. It has two different types over here they have mentioned, but they have basically broader categories. Chromoplast and the leucoplast. In chromoplast they have talked about the chloroplast. But we know when we generally study, it has three types. Right? In your textbook they have mentioned two, but when we talk about it, we always say it's three types: chloroplast, chromoplast and the leucoplast. So the chromoplast contains the pigment chlorophyll. Now they have just, as I mentioned, they have just clubbed it over here. They contain the pigment, different colors of it, right? We have the chloroplast, which have the chlorophyll pigment, which gives a green color. Then in the chromoplast we have xanthophyll and carotenoid, which gives the beautiful colors to the plants and vegetables. And then we have the leucoplast, which is white or colorless plastid, which we usually see in the roots, right? It actually plays a very important role in the storage of oil, starch, etc. Very good. So here they have mentioned about the chlorophyll, they have mentioned about the yellow and the orange pigments also. They have mentioned about the leucoplast, right, which is there for the storage, stores starch, oil and protein granules. So directly the information can come from the textbook itself. Three things it is storing and we can write 3. Are we clear? Yes? Very good, very good. CCL, yes. Chloroplast, chromoplast and leucoplast, very good. So of course we talk about the chloroplast specifically, right? They have mentioned that they have numerous membranes, right? And of course we call them as the stroma, right? They are there in the stroma. And they have thylakoids and they have the small thing which is the thylakoids as disc-like structures. And they have the chlorophyll. Yes, are we clear, everyone? Okay, Harish sir, can you put me back to the PPT mode? Just, yes, I'll come over here, yes. So over here, we're talking about the chloroplast, right? So chloroplast have chloroplast, right? We have the double membrane, right? And of course in that we have these disc-like structure. Now these disc-like structures are called as the thylakoid, right? And inside these disc-like structure we have the chlorophyll, right? And the stack of these is called as the grana. And outside we have this, we call it as the stroma. Okay, so you can check more about it. We have a detailed session on it and we can discuss about it. Great. Okay, moving back to the textbook now. here. Yes. Now that's about the plastid. Everyone clear? Yes, stroma right over here. If you can see, journey to become a doctor. Stroma is nothing but the surrounding that we have outside the thylakoid or the grana, right? Basically that space inside the chloroplast. We call it as a stroma. Are we clear? Very good. So plastids are the, we call them as the kitchen of the cells, right? Of course they are the one. We don't call them as the kitchen of the cells, chloroplast is the one. Now plastids are present only in the plant cells and they are the one which are of three different types. We have chloroplast which have the green color pigment, chlorophyll, and that actually help in making the plants their food. We have chromoplast which gives a colored pigment to the plants and vegetables and to the flowers, fruits, right? And then we have the leucoplast which have white color or colorless, and they actually help in the storage of the proteins, protein granules, oil, starch, right? Yes, Purnima, respiration occurs in mitochondria. So are we clear with this? Everyone quickly? Yes. Very good, very good. Are we clear with this, everyone? A quick thumbs up. Very good. And here we go, right? And here we go to the vacuoles now. Now vacuoles are the storage sac for liquids and for the solid. Vacuoles are small size in animal cells and they are big in the plant cells, right? In plant cells we'll see a big vacuole, but in the animal cells we'll see a small vacuole. The number of vacuoles will be more in the animal cell as compared to the plant cell, right? The central vacuole in some plant cells occupies up to 50 to 90% of the cell volume. Then in plant cells, they are saying that they are full of the cell sap. Now what is the cell sap? Anyone? Thanks everyone for the thumbs up. Anyone, can you tell me what is cell sap? We have discussed about it, right? What is a cell sap? Cell sap, it's a fluid, right? Just imagine in a vacuole, we have the cell sap. Now, what will be there in the cell sap? What are the components of it? Quickly. It's a fluid for sure. This will be having the lot of nutrient, right? Yes, very good. Very good, Anshu. Yes, very good, everyone. So, of course, the cell sap will include the lot of nutrient, right? Of course, a few waste materials also, right? And basically, it's more of fluid which is there, and it will be providing the turgidity and rigidity to the cell, right? So, now if we have let's suppose we have this water bottle, right? Inside it we have a fluid, right? You can see what we have inside this bottle. We have the water, right? But of course, if we take this water outside, this bottle I can easily crush it, right? Yes or no? Oh, look at it, has a green cap, but on the screen it looks different. Nice. Yes. What do you think will happen to the bottle? If I remove the water from it, it will not be able to provide the turgidity that it has. I can easily crush it, right? But when it's there, it's put the pressure also. So similarly, the vacuole also has the fluid, right? And it just puts the pressure and gives it turgidity and the rigidity to the plant cell. Very good. Okay, so many important important molecules are stored in the vacuoles, and these fluids includes amino acids, sugar, proteins, right? And in single cell organelles like amoeba, the food vacuole contains the food items that the amoeba has consumed. In some unicellular organelles, specialized vacuoles plays a very important role in expelling the waste and the excess material out. Yes, turgidity means stiffness. In a short way, if I have to tell you, right? So let's suppose this is turgid, right? Like if I just, even if I put a lot of pressure, it will not be getting crushed. So turgid in a very layman language say it's very stiff, right? Yes, are we clear everyone? In the plant cell, the vacuole is large, that's why the nucleus could be, yes. Yes. Very good, very good. So with this we are done with the vacuole, right? Now there's a few more lines about the vacuole that we have over here, which is that each cell does have an acquired structure about the ability to function because of the various different organelles that we have, right? And these organelles help them in performing various activity, including the nutrition, digestion, excretion, and whatever that happens in the body. Now the cells, of course, plays a very important role in maintaining the life, and that's why we call the cells as the structural and functional unit of life. So by the end of this all, we can say that cells are really very important. Yes, okay. Turgidity and rigidity are very stiff, very hard. Yes, a kind of toughness that it provides. Are we clear everyone? If someone asks you, right, that why the cells are the structural and the functional unit of life, now I'm sure you have the answer for it. You can say they help in performing lots of function. And each of these functions make sure that all of us are alive and working. Very good. So we are clear with this, right? Phagocytosis, to be more specific, right? Yes, phagocytosis happens in the amoeba. Very good. Now comes the last part, which is not that much in detail in your textbook, that is a cell division. So they are talking about the cell division. So new cells are formed in the organism in order to grow, to replace, or to basically to help in the formation of the gametes, right? That's why the cell divide. Now we have two types of cell division. We have mitosis and meiosis. And what is cell division? The process by which new cells are made is called as cell division. Yes, so let's quickly discuss about mitosis and meiosis. So the process of cell division by which most of the cell divides for growth is called as mitosis. Now for example, in our skin cells, right, what we have the cells, right? Now these all needs to divide, they need to repair themselves, they need to grow. That's why they have a type of division which is the mitosis, right over here. If we go over here, we can see this is the mitosis, where the cells will make the identical copies of themselves, right? It will be equational division or the equal division. Yes, the number of chromosomes will remain same from the parents to the offspring. Journey to become a doctor, we have done a detailed session on the cell division. In this session, I will not be explaining you about that, right? Basically, it's a phase that we see in meiosis, right? One, and that's a very detailed process we have. But we have done a video, you can go and check that out. Yes. Very good, yes. So in mitosis, no chromosomes will be getting changed, right? Number will remain same. And we usually see such type of divisions happening in our body throughout. Only at one place we see the meiosis happening. And that we see in the production of the gametes, the formation of sperm or the ovum. In that what happens? Let's quickly see. In meiosis, what we'll see the reductional division. The chromosome number will become half. For example, in humans we have 46 chromosomes. In the daughter cells, we will have 23 chromosomes. From 46 to 23, right, we know that the number is getting reduced to half. 46 over here is diploid, right? And 23 over here is the haploid. So remember that. Everyone, are we clear? Yash, this week you will have. Awesome. Are we clear everyone? Haploid and diploid. So now let's quickly read from the textbook. So we have in this process, each cell is called as mother cell and they divide to form the two identical daughter cells, right? We're talking about the mitosis over here. The daughter cells will have the same number of chromosomes and the mother cell, and it actually help the organism to grow and repair the tissues. Then over here what we have, that specific cells of the reproductive organs or tissues in animals and plant cells divide to form the gametes, which after the fertilization give rise to the offspring, and they divide in a different way where the cell will be dividing 2 times, and what we'll see, we will see that there will be the formation of 4 new daughter cells, right? And in the mitosis we'll see 2 daughter cells formation. Yes, are we clear? Yes, and of course the new cells will have the half the number of chromosomes. Yes, are we clear everyone? Yes, quickly give a thumbs up. Are we clear with this? Tejaswini, reductional division means when the chromosome numbers are getting reduced, right? That's why we call it as a reductional division. Rafiq, we have done a detailed video on this bache. I know that this is a very short revision of it. In the CBSE, there's no much details of it. They just mentioned the cell division, mitosis and meiosis. Mitosis occurs in the body cells throughout the body, including the skin, right? Where of course the parent, mother cell, will produce the 2 daughter cells with the same number of chromosomes. Meiosis occurs in the reproductive cells, right, that we have, and we will see the formation of sperm and the ovum, right? Sperm and the ovum. Now, of course, there we'll see the reductional division. The number of chromosomes will get reduced to half. So that's a very brief information we have on the cell division over here. Rafiq, if you want to learn more further, there are videos that we did in great detail, so we can see from there. Yes, with that we can say we are done with this chapter. Now, I'm not asking anything big from you guys. After this class, please just go back and now you sit quickly and read all about the organelles. Yes, it's a very simple session. We're moving back to the slides now, and let's just quickly move over here. Yes, so do that and you will be amazed, and it's really very easy. Awesome. So with that, we will end our session. Yes, we'll end our session. Okay, wait. Okay, okay. Wait. Yes. So you're asking me to explain you again about the mitosis and meiosis. Okay, last time for everyone, because I understand this is a very difficult topic. Mitosis and meiosis. Now everyone, I'm just explaining for the like what we have in CBSE. We're not going in detail, right? Mother cell will give rise to the 2 daughter cells. 2 daughter cells. Now in this, mother cell will give rise to 4 daughter cells. Second point for all of us to remember is that chromosome number will be same, right? Chromosome number will be same, and over here chromosome number will be half. Are we clear? Paul, if you focus over here, you will get the answer. Then the important thing that we have to mention occurs in body. Specifically, we will mention few examples like skin cells, etc. And it, okay, this pen and the screen. Yeah, it occurs in the gametes formation, or we can say the reproductive cells. Occurs in the reproductive cells. Okay, so that's one thing you need to remember about these 2 important processes, meiosis and the mitosis. Very good. Well, with this everyone, we are kind of done with our session. If you've enjoyed our session, please don't forget to hit the like button for the video and subscribe to the channel. Yes, and of course, if you haven't joined the community, please join this week on Friday. Right, I'll be coming live on Telegram, so we'll have a quick small discussion. So right, this Friday everyone on 26th of August, right? That's 26th of August. At 3 p.m. I'll see you on Telegram. Yes. Paul, in this particular chapter, there is nothing much for you to learn in terms of the diagram. You can just practice with the cell plant cell and the animal cell. And you know that we have got you covered, everyone. Hit the like button and share with your friends and subscribe. Right? Yes. Okay, Devdas, but yeah. Okay, I'll not say anything. But yeah, it's up to you. Tejaswini, you were asking about the homework question. I think that your exams are going on and all of you are learning for the examination. That's why I'm not giving any homework. As of now, we will have the homework question later, but I have given you the homework question indirectly. Read the book, right? Read whatever we have read today in the class. So that will be the homework. Yes. Yes, Paul, you can learn more about it. Yes, it's a live session. And with that, everyone, lots of love to each one of you. We'll be meeting really very soon. That is tomorrow. We'll meet again. Right, take care of yourself and keep on learning with BYJU'S. Bye-bye.